Great. Thanks, Shannon, for that introduction. So I just want to welcome everyone and really thank the Vanderbilt University Institute of Imaging Science for giving me the opportunity to speak to you guys today. So as was stated, my name is Adam Bush, it still is. And I'd like to tell you about ironing out the kinks, new methods and lessons learned in translational MRI. But before I start, I'd also like to thank Shelly Kraninsky. So we met at a renal conference maybe 6 months ago, and we really hit it off just talking about where the field of renal imaging is going. So from that perspective, I think to all the trainees out there, go out there, network, it should be fun. And you never know where it might take you. So yeah, I'll go ahead and start. Okay, so as was stated, my name is Adam Bush. I'm a new assistant professor at the University of Texas at Austin in the biomedical engineering department. And I think what makes me interesting or unique is that I was actually a PhD engineer, but I was trained by two clinician scientists. So first, pediatric cardiologist John Wood at Children's Hospital Los Angeles, and more recently, pediatric radiologist Shreyas Vasanwala at Stanford University. So I think that training really informs the type of questions that I'm interested in asking, particularly those related to translational medicine. So how can we get the tools that we develop to really use in clinical medicine? And I think that really underpins what it means to be a biomedical engineer. So when we think about biomedical engineering, I think it's this combination of engineering or math and physics with the study of life sciences like physiology and biology, and of course, to be used in clinical medicine. And I think that is what translational medicine is, often referred to as bench to bedside medicine. It's the injection of math and physics, understanding about life sciences into clinical medicine for the improvement of outcomes. And I think probably the most quintessential or the most prototypical translational field is kind of pharmaceutical development. But the thing about pharmaceutical development is it's actually extraordinarily costly. So if we think about developing a single pharmaceutical, it usually costs about $1 billion to develop and takes anywhere from 10 to 20 years to actually meet and get into the hands of patients. So if you were like me, I didn't get paid $1 billion for my stipend as a grad student, and I definitely didn't want to be in school for 20 years. So I think these type of projects don't really lend themselves well to individual scientists working on them. But that's just because they're very large projects. And if we think about it, it's because it has to do with the translational pipeline. So typically in pharmaceutical development, there's this kind of target that is generated, and then it has to be demonstrated to be both safe and effective, typically in animal models. And then from there, it moves on to human studies in phase 2 and 3 trials, where we test the safety and efficacy. And this takes many years to do. And also at the end of it, it then has to be profitable. So there has to be a large enough population that is willing to buy the drug to be used. So that really creates like a problematic pipeline, particularly if there's any errors that come up or any hangups along the process. So that essentially means that the whole pipeline has to get shelved, and it makes drug development extraordinarily expensive. However, as an imaging scientist, we're actually at a huge advantage here. Like it's ginormous, because effectively what happens is the pipeline is removed because imaging modality, particularly MRI, is already demonstrated to be safe in humans. That means that anything we develop, our only task is to demonstrate that it's effective. If it's effective, then it can almost immediately be used in patients. And we don't have to necessarily even demonstrate it's profitable. It just has to add value. So how do we add value to things? We can add value in terms of making scans faster, making them safer, making better images. There's a lot of ways of adding value that don't necessarily translate to profit. So imaging is, quite frankly, the best translational field to be in if you really want to improve patient outcomes. Furthermore, I guess if you look at this kind of linear model to bench to bedside translational medicine, imaging allows us to kind of turn that on its head a little bit, because now, since we can develop things in humans, we can have this whole translational pipeline that is kind of all communicating with itself as it works together. So for my work, I focus on MRI development techniques particularly around blood flow and hemoglobinopathies disorders, and particularly in children and those that are disenfranchised populations. And by studying all of these things simultaneously, I think it just expedites the translational process. So when we think about the challenges associated with being a translational scientist, I think they're threefold. So anytime we develop a technology, we have to ask ourselves, is it meeting these three kind of criteria? And you can think of them as the wrinkles. So we have to make sure we handle these wrinkles. The first one is, do we have a clearly defined clinical use case? So doctors need to know when and where to use a technology in order for it to be used or be effective. Second is, does the technology provide some type of objective clinical support? So doctors not only need to know when and where to use the technology, they need to know how the knowledge that they gain from that technology should inform their clinical decision-making. So do I treat if it's above this level, or do I lower the medication if it's below another level? That's really important information for a clinician in order to use a biomarker in the clinic. And then lastly, of course, it has to be reliable in both healthy subjects as well as patients with disease. So oftentimes as engineers, we might find ourselves validating things in graduate students. However, in order for it to really to be used, it has to go to the next level and really make sure it works in the patients who are going to need it. So for my work, I really work on these iron-based imaging approaches. So these iron-based imaging approaches can really remove these wrinkles. So as far as the rest of this talk, I'm going to give you a historical example of how this kind of framework works, and then we're going to go over some more of my own work on how it contributes to this using this pipeline. Okay, so let's talk about a little physiology. So as stated, my work really focuses on cardiovascular disease, and particularly around blood and blood imaging. So blood is this colloidal mixture of plasma and red blood cells, where red blood cells are kind of just these like sacs of hemoglobin, where hemoglobin is the oxygen carrying protein that carries oxygen throughout our body. And the hemoglobinopathies are therefore now these genetic disorders associated with hemoglobin production. So essentially there are the structural variants like sickle cell disease, and there's also the coding variants like thalassemia. So historically, if you had one copy of these homozygous recessive diseases, you would have protection against the plasmodium responsible for malaria. So that's why they're still around today, because if you're a carrier or a trait for these conditions, you have protection against these very deadly diseases. Unfortunately, however, if two carriers have a child, there's a 25% chance that they'll have this debilitating disease. Historically, these children probably would have died very early in childhood, so they weren't really selected for. However, with modern medicine now, these children are living well into adulthood, and now we have to take care of them in the medical system. So if we look at where these hemoglobinopathies occur, they actually occur in the same places where you see malaria, so, or in other kind of blood-borne illnesses. Okay, so what's the medical approach here? So in terms of thalassemia is this genetic mutation that prevents the production of hemoglobin. And it typically doesn't kick in until after about 1 month or so, because as a fetus, we actually produce a different type of hemoglobin known as fetal hemoglobin. And it has a higher binding oxygen, higher affinity for binding oxygen, so we can remove oxygen from our mother's blood. However, if you have thalassemia, that means once your fetal hemoglobin genes turn off, you don't produce any hemoglobin, which is problematic. So these patients are given blood transfusions from a very early age, and that's what's required for them to stay alive in many cases. And though that's a life-saving kind of technology or treatment, it does have some drawbacks. So one of the main ones is something called iron overload. So we don't have a natural mechanism for excreting iron from our bodies. We never needed one kind of in prehistoric times. So what happens is we'll actually accumulate all that iron from the hemoglobin that was getting injected into our bodies if we're on blood transfusions, and it'll build up in the solid organs of our body, particularly things like the liver, the heart, the pancreas, and it's toxic in high doses, so it'll actually cause organ failure. And when your heart fails, you typically die. So this is problematic. Fortunately, there are these therapies known as chelation therapy, which can remove the iron. It binds to, it's a drug that'll bind to iron and then be excreted in our stool. However, of course, in order to give this drug, you need to know how much iron is present. So typically how this was measured in the past is you would have something called biopsy, where pieces of the liver are removed and then they're measured for their iron content via microscopy and other techniques. Unfortunately, biopsy, of course, is painful. No one likes pieces of their flesh being removed. Additionally, it's not very accurate. So the liver is a massive organ, and it's prone to sampling errors if you just take off one sample for biopsy. Additionally, it doesn't really allow for the measurement of iron in the heart, because even biopsies of the heart just a single time can be fatal, because and we don't want to do diagnostic procedures that can be fatal like that. So it's typically avoided. Okay, so what are the engineering solutions here? So fortunately, in the early 2000s, late 1990s, some really bright scientists and clinicians decided, wait, we're trying to measure iron. How about let's use a giant magnet? So turned out that approach actually worked, that we're able to quantify iron with these relaxation parameters known as R2 and R2 star. And that was some of this early work. And it was completely game-changing for the treatment of iron overload. And I think that's really solidified in this graph of mortality in the UK, thalassemia patients by decade. So if you'll notice here, we see that in the 1960s, this is when the first patients with thalassemia were given blood transfusions. And then from there, you'll notice that the bars are getting bigger, meaning patients are actually surviving. Unfortunately, you'll also see these bars getting bigger in the red, showing that they're dying from iron overload. And though chelation therapy first came out in the 1970s, it wasn't until here in the early 2000s where we see this ginormous drop in those red bars, meaning patients stopped dying from iron overload. And that was the exact same time when MRI started being used to measure iron. There's no coincidence there. So now it's patients that are well managed for their iron therapy, it's just, they don't die from iron overload anymore. It's a kind of a solved problem. So I think this is a great example of how just a relatively simple imaging strategy can really inform clinical medicine to really allow us to improve patient outcomes in a very meaningful way. Okay, so how does our kind of framework for assessing a biomarker or iron-based imaging approach work here? So first, is there a clearly defined clinical use case? And of course there is, it's to identify iron, so that's great. And then does it provide objective clinical decision support? And fortunately it does. So some of the early data that came out following these MRI studies was the outcomes data. So here I'm showing some of that data showing that here's heart R2 or T2 star, and what you'll notice is when the T2 star drops below 20, you see this precipitous drop in ejection fraction, which is essentially a marker of heart function. And this is kind of the information that doctors need in order to say, okay, how can I use this biomarker? Essentially what it's saying is like, don't let the T2 star in your heart drop below 20 or you'll have problems. So that's great. It provides objective clinical decision support. And then lastly, is it reliable and easy to use in both patients and disease? And in fact, it is. It works across the entire R2 star range, more or less. So many people would consider this a perfect biomarker. However, there are some challenges still associated with iron imaging currently. And this is where I'll kind of move on to some of the work that I've worked on. So some of the ongoing challenges with iron imaging are particularly related to physiologic motion. So motion in MRI will corrupt our images, as seen here. Just normal respiratory motion will make our T2 star maps almost unusable. So typically what is performed are these gating strategies like cardiac or respiratory gating, which can be used to essentially freeze the motion in one motion state. Unfortunately, these gating strategies have several drawbacks. First, they can make long scan times. They can be prone to failure if the leads fall off or the bellows stop working properly. Additionally, they're not always the most comfortable things to be using. And some patients can't even use them, especially children. So anesthesia is also commonly used in many cases. And that's adds additional risk. So a great value add here would be if we can make a motion robust strategy for doing these kind of relaxometry-based measurements. So in order to understand how we do that, let's just do a very simple physics review. So once we excite our signal in MRI, it'll decay with this model exponential defined by this relaxation constant known as T2 star. We can then sample images at different echo times and then do a fit to get a T2 star map. And typically when we acquire images in MRI, we actually sample our image in this frequency domain or this K-space representation, which is related to the image by a Fourier transform. So when we sample in multi-echo imaging, typically what happens is we raster back and forth a single line of K-space. We do that several times. So, and say if we're doing cardiac imaging, we might do a set of K lines per cardiac phase until we acquire all of K-space. The problem here is that if there's any residual motion during that acquisition, we can get these aliasing or ghosting artifacts. So we know, however, though, there's some well-vetted strategies for reducing motion artifacts like non-Cartesian imaging. And essentially how it works is because it oversamples the center of K-space and also has diffuse kind of motion or diffuse aliasing artifacts that look kind of like noise, we're able to denoise them, which allows us to generate these relatively motion-free images. However, it's not really performed very often for multi-echo imaging. So here, the kind of impetus for this was, how can we do multi-echo imaging with non-Cartesian? So the thought was, if you do this kind of looping pattern in K-space, you can acquire multiple echoes, and then you can rotate that trajectory until you fully sample K-space, and you can get multiple echoes that way. And turns out this kind of looping trajectory was first described by Doug Knoll, probably over 20 years ago, and it's called a rosette. So I was inspired to use a rosette in order to do this multi-echo imaging. Okay, so in this approach, I take a single rosette. It has 5 petals. That gives me 5 echoes. I can then rotate that rosette by the golden angle, 137 degrees, so I can fully sample K-space. Then I can break that trajectory up into individual echoes and then perform reconstructions for each one of those echoes so that I can fully sample K-space. And it turns out that this acquisition strategy is actually well suited for compressed sensing model-based reconstructions. So in compressed sensing, we try to minimize the difference between our data that we acquire and the image we're trying to reconstruct in this kind of iterative fashion by imposing constraints on what the image could be. So each time we iterate, we'll reduce some of the noise-like artifacts, which eventually ends to us having this like very motion robust or denoised image at the end. And fortunately at Stanford, I was able to work with many experts in compressed sensing, also at Berkeley, and really leaned on them heavily for those aspects. But additionally, there are online tools that will allow you to actually reconstruct images with very fancy reconstruction algorithms with just using online tools. So BART, or the Berkeley Advanced Reconstruction Toolbox, is one such tool. So using those tools and that acquisition strategy, I'm able to now reconstruct these rosette images for multi-echo imaging. So here I'm showing an example image of a standard Cartesian multi-echo single echo image right here, and then the corresponding T2 star map. And then I'm showing an image of the similar time span image from a rosette acquisition. And what you'll immediately notice is that the myocardium blood pool delineation is much clearer in these rosette acquisitions during breath holding. And then, of course, during free breathing, which is important in some patient populations, whereas the standard technique will now have additional artifacts in the liver as well as the myocardium, the rosette acquisition is just largely robust to those type of measurements. Okay, so we can then perform reader studies where we can quantify this image quality in some meaningful way. And in all the cases, the rosette outperformed the Cartesian strategy. So we see in breath holding, free breathing, and failed breath holding, the rosette images look relatively similar, though like the Cartesian images are largely corrupted. As far as the accuracy, we can now compare the accuracy of this technique to in a phantom, where we can use ferromoxitol, which essentially is an iron-based contrast agent. And we can show that the 1/T2 star, the R2 stars, perfectly correlated with the iron concentration. And then, of course, because we're working in a completely safe, a non-invasive technology, we can actually use this immediately in patients. So here we're now showing the Cartesian in healthy volunteers, but then several patients with actual iron overload. So we're able to show that the correlation and accuracy is actually quite good in the liver. It's not so good in the heart, but this is almost certainly due to limitations of the existing technique, because you'll notice the Cartesian strategy just doesn't give the greatest myocardial delineation in the heart, especially in the healthy range. Okay, so to date, this sequence has been used over 500 times. So that means at Stanford Children's Hospital, that means less sedation. That means that clinicians are more confident with the decisions they're making. And this really just shows how doing relatively minimal amount of work, this didn't take 20 years of development, we can already make meaningful impacts on patient outcomes. So this is kind of really the power of imaging. And of course, with any working kind of product, there's always offshoots. So I've been fortunate to work with some really brilliant summer students and as well as grad students. So Shreya Ramasandran worked with me as a summer student at Caltech. She's now a third-year PhD student in Dr. Mickey Lux's lab at Berkeley. But she helped work on choosing the right shape of the rosette by doing some simulation modeling. Additionally, Suma Anand, she's now a fourth-year EE student in Dr. Mickey Lux's lab, worked with me briefly looking at fat spectroscopy using rosettes. And then, of course, Cynthia Chen, who is also a summer student, and she's now, I believe, third year at Caltech in the CS program, looked at how can we begin to work out some of the gradient timing corrections that are needed for going very quickly when sampling case space. So here we're looking at reducing gradient delay, boost errors in some of these rosette-based techniques. Okay, and then of course, single slice is great, but how can we get volumetric coverage of the heart? So we're beginning to now move into that space of just doing a multi-slice approach to doing cardiac imaging. And of course, the next thing you realize is as soon as you can do cardiac whole heart coverage, why can't we just resolve the motion of the heart itself? So here we're showing some preliminary data, just showing that yes, you can actually even resolve the cardiac motion, which will be useful for quantifying things like heart R2 star as well as heart function simultaneously. But of course, these images are highly undersampled, so I'm still working out the best way to proceed on projects like that. Okay, so now I will change gears a little bit and talk about other aspects of translational work. And for this, I now want to talk about another hemoglobinopathy known as sickle cell disease. So sickle cell disease is the most common genetic genetic disease in the United States. It affects about 100,000 Americans, primarily of African descent. And what happens is it's caused by an abnormal hemoglobin molecule that will polymerize, essentially form these like long rigid fibers when it becomes deoxygenated, and those fibers within the red cell will lyse the red cell, meaning they'll like split it open, as well as they can block and occlude blood flow. So of course, blocking blood flow in organs like the brain is pretty devastating because it can cause stroke. So about 11% of patients below the age of 18 will actually have a massive stroke, making sickle cell disease the most common cause of pediatric stroke if left untreated. Okay, so unfortunately, there's also not only massive stroke, but there's something called silent stroke. So silent strokes are called silent because there's no real neurological signs and symptoms like would typically be associated with like hemiplegia or aphasia from a major stroke. And they occur in about 50% of children by the age of 20. So essentially, almost half of these children have these small infarcts that are only only seen on MRI images. Okay, and though they're considered benign, I think this representation of a healthy teenager as well as a teenager with sickle cell disease that's deemed healthy shows that this is not really a silent disease. Okay, so currently the biomarkers that are used to detect stroke risk in sickle cell disease is transcranial Doppler. And this is where an ultrasound probe is placed on the middle cerebral artery of young children. And if the velocity is above 200 centimeters per second, then they're deemed to be a high risk for stroke. And it's been shown that if you place them on chronic transfusion therapy, they'll actually reduce their stroke risk by 90%, which is fantastic, right? Strokes are bad. We want to prevent those. However, there are some drawbacks here. First of all, we're using ultrasound transcranial Doppler, which is effectively like a 40-year-old technology. So that definitely needs some updating. Additionally, about 80% of the patients that would have gone on to be put on chronic transfusion therapy are not going to develop a stroke. So that means there's an 80% false positive rate. And chronic transfusion therapy for life is not a benign therapy, meaning that many of these patients will now have to receive a blood transfusion at a hospital every 2 to 3 weeks for the rest of their life. So that means you miss a lot of school, miss a lot of work. There's, of course, the risk of alloimmunization as well as iron overload, like we talked about. So if we could avoid that, that would be preferable. And of course, transfusion therapy is shown to not actually reduce the frequency of silent stroke progression. So we could do better. But how can we do better? This is where maybe we can develop some de novo biomarkers that could be predictive of stroke risk. So if we use our kind of framework for how do we develop a biomarker here, first, do we have a clearly defined clinical use case? I think yes, it's to identify stroke risk and potentially decrease the frequency of stroke. But then what about the objective clinical decision support? We don't actually have any data showing that when or where we should be using these biomarkers and how it affects outcomes. So we really need to measure that. So first we need to decide on what biomarker to use. But where do we start? Well, one of the places to start might be with oxygen utilization. So the brain is an interesting organ in that it only weighs about 2% of the mass of our body and consumes about 20% of the oxygen. So it has an extremely high aerobic metabolic rate. And so maybe looking at oxygen utilization locally would be a logical place to start for biomarkers. Unfortunately, oximetry is actually quite easy to use or developed in MRI. So oximetry essentially is just how do we differentiate oxygenated versus deoxygenated blood. And I think intuitively we know that there's a difference between oxygenated and oxygenated blood based on just visualization. So we know that their absorbance spectrum is different. So we'll see here that deoxyhemoglobin has a higher absorbance of red light, and that's why oxygenated blood appears red, because it's actually reflecting the red light back at your eyes, so it looks red. And contrary to popular belief, venous blood isn't actually blue, it's just a very dark red color. But that allows us to differentiate blood optically. But how can we do this magnetically? So it turns out that this is actually this is a profound property of blood, is that it actually behaves quite similarly magnetically. That if you place blood in a magnetic field, oxygenated blood is what we call diamagnetic, so it actually will oppose the magnetic field, whereas deoxygenated blood is paramagnetic. It'll actually enhance the magnetic field. And we can use that as a basis for being able to differentiate venous structures from arterial structures. Also, it's the basis of something called fMRI, probably one of the most important developments in MRI in the last 30 years. So how fMRI works is we use typically some type of activation in the brain, like this blinking checkerboard, that then causes some local activity of neurons that begin to fire. There's now a vascular response, which typically is higher than the increase in metabolic rate. So now there's more arterialized blood going to that region. And now we can detect that on an MRI using T2 or T2 star weighted, as well as other techniques. So that allows us to now localize where exactly this blood flow response is happening in response to function. So one of the things that we were interested in is looking at how can we kind of change the model a little bit, and instead of using activity-based, how can we look at the vascular response in response to hypoxia? So in order to do that, we delivered 100% nitrogen to subjects, which is effectively displaces the oxygen for about 20 seconds. This causes a very acute drop in their peripheral arterial saturations. We can then look at the vascular response in response to that acute challenge, and with bold imaging. So you can see a drop in the saturation, and then you can look at where the vasculature is kind of reacting. So we did that in patients with both silent infarcts and patients, and they actually had very different behavior in response to that hypoxic stimulus. So here we're showing the kind of group-wise averaging of patients that had silent infarcts and those that did not. And what we can show is that the patients that have infarcts have a much more heterogeneous response to this hypoxic stimulus in both their kind of time to peak, how fast they're responding, as well as how long they're responding to that challenge, which might be able to give us some information about what is happening on a microvascular level. So it seems like there's much more microvascular heterogeneity in patients that have silent infarcts. But I think what's probably even more interesting is that one of the things that came out of this work is that we realized that, wait a second, maybe we can use this challenge as a way of actually measuring blood flow in a non-contrast repeatable way. So if you think about it, this hypoxic stimulus is causing a bolus of deoxyhemoglobin, which is paramagnetic. So very similar to an injection of gadolinium, where you would do a first pass perfusion study, where you would inject gadolinium into a vein, and then you watch as the contrast passes through, and then you use kinetic tracer modeling to extract these hemodynamic parameters. You can actually do the same thing with just breathing in nitrogen. There's no needles, no injections, no allergic reactions. It is 100% repeatable. So this is actually something I'm really excited about, and I think has legs in the future of MRI. Being able to measure blood flow in any organ in the body without using gadolinium. So we're then able to use those kinetic tracer models to make these very elegant pictures of hemodynamic parameters. So here I'm showing blood flow maps, transit time maps, and blood volume maps and controls in patients with sickle cell disease on a population level. And what we're able to show is that the predictions are more or less aligned with what we'd expect. We see higher blood flows, we see shorter transit times, larger blood volumes in patients with sickle cell disease. But of course, this is very much so in early days for this technique. So it needs to kind of be democratized, and I think more people should be using it because it's awesome technology. Okay, but the problem with that strategy is if we remember our 3 criteria, it's not particularly easy. So doctors like simple solutions. They like numbers, like one thing. They don't really like to look at maps in order to determine how to act clinically. So how can we come up with a number that is suitable for the risk of stroke? So for this, we can use the Fick principle for looking at oxygen consumption. So we know that oxygen utilization is simply the difference of oxygen inflow to the brain minus oxygen outflow to the brain. And fortunately, MRI is actually fantastic for measuring blood flow. There's a number of different techniques that can be used to do that. And fortunately, there's a consensus in sickle cell disease literature showing that oxygen delivery is elevated due to their level of anemia. And we have several publications looking at that, but it seems like the white matter is particularly at risk for ischemic injury because it's not as elevated as the gray matter is. But the challenge now is how do we measure venous outflow? And we have a flow measurement, so we really just need to measure the oxygen saturation. So how do we measure oxygen saturation in the brain? So one way of measuring oxygen saturation in the brain is this technique known as T2 relaxation under spin tagging, or TRUST. So in TRUST, how it works is we use an arterial spin labeling tag in the brain. We can then allow that blood to flow to the sagittal sinus and then make these T2 measurements at different effective echo times. And essentially what we can do is now we can have a completely isolated blood pool that we're now making T2 measurements. And similar to how we described before, we know that there's this relationship between the amount of deoxyhemoglobin and this relaxation parameter T2. So we can now use these empirically generated calibration curves in order to get at what exactly the T2 is in the sagittal sinus. And these empirical calibration curves are largely based on spin exchange models or Lewis-Meibum exchange models. And though they might look complex, they're really quite simple because most of these parameters are either sequence parameters or properties of blood. So largely what is done in the literature is we can reduce all of those parameters into these coefficients that will describe these relatively basic relationships between the amount of deoxyhemoglobin present and what we should see in terms of relaxation or R2. And that's how we generate these empirical calibration curves. Okay, but it's an open question whether these calibration curves are actually reliable and easy to use in patients with disease. So we wanted to explore that a little bit more. So here, we weren't the first people to do trust, and arguably we're not the first people to do trust in sickle cell disease. So there's actually been several publications looking at trust in sickle cell disease patients, and the general consensus prior to validation work was that that cerebral extraction was actually elevated and metabolic rates were elevated. The problem with that early work is that it had several limitations, primarily that the calibration equations were defined in cow's blood, where cow's blood has about the erythrocyte size, or red blood cells are about half the size of normal human blood cells. Additionally, the range in which that early work was defined in terms of hematocrit was a relatively normal range, so didn't expand into the anemic range. So some of my earlier work tried to overcome those limitations. So we would actually make measurements of these relaxation parameters in healthy hemoglobin A containing blood, and we could compare it to our bovine model. So here we're seeing the bovine models that are typically used, and here we're showing the hemoglobin A model that I generated using ex vivo data. And what you'll see is they're more or less are the same in shape. However, if I plot the bovine model over the same hematocrit range, you see this large nonlinearity that happens in the anemic range. And this essentially just contributed to errors because it wasn't defined over that region. So any nonlinearities really just took off in that anemic section. So that solves some of the problems. However, it doesn't address the question of these calibration equations actually work in patients with sickle cell disease, because they don't actually have normal blood. So once again, we made blood measurements inside the scanner. So here's one of our earlier setups, just showing that these were very controlled experiments where we controlled for things like the sedimentation rate, the temperature, oxygen levels, hematocrits, in order to generate these calibration curves. So here is the hemoglobin A model that I derived, and here's the hemoglobin S model, and they are distinct. There are some interesting features that are different about them. So one of the things is that the oxygenation dependency for this hemoglobin S model is much steeper, as well as the hematocrit dependency wasn't as strong. There was other effects that were larger than the hematocrit dependency in this model over this narrow range of hematocrits that we measured. But we're not only interested in what the relaxation of blood is, we're actually interested in what are the predictions about cerebral physiology. So if we look at the old bovine model kind of physiological assumptions, we show that the venous saturations were much lower and the accompanying cerebral metabolic rates were much higher in sickle cell disease patients. However, if we use these more vetted techniques for sickle cell disease populations, we get the complete opposite behavior. We see that patients with sickle cell disease have much higher oxygen saturations, and they have a correspondingly lower oxygen consumption in their brain. So this was a really profound but contentious finding, because of course, anytime you are at odds with the literature, then there's always room for conflict there. So fortunately, since this work has been published, there's actually been additional work that's been done. So another group at Johns Hopkins has now validated that yes, in fact, sickle blood does have steeper relaxation with respect to oximetry. And now a calibration curve between all of us has been it's like a consensus calibration curve has been generated. But additionally, we've now been able to do studies in patients that have normal containing hemoglobin A blood. So there is no argument about their calibration curve, but they're anemic. And what we're able to see is that they have the same physiological response. We see decreased oxygen metabolic rates in their brains just by being anemic alone. So that kind of gives support to the idea that what we're observing with these calibration curves in sickle cell disease is actually correct. Additionally, we can look at the historical literature now with invasive techniques in anemic populations and show that, in fact, the historical literature shows very similar curves to what we're able to generate in terms of the more anemic you are, the lower your metabolic rate is. Okay, but why would hemoglobin S containing blood have a different relaxation? Well, if we look at these Lewis-Meibel exchange models, one of the parameters in this model that we just concatenate into a single coefficient is actually the shape of the dephasing object. So since it's well known that these patients with sickle cell disease, when their blood is deoxygenated, that they take on these very strange shapes, it stands to reason that that's likely why the blood is experiencing these different calibration curves. Okay, so how do we explain the difference in metabolic rate and oxygen extraction? Well, our working hypothesis is that of vascular shunting. So in a normal vasculature, blood will enter to the vein or the artery or arterial, and it'll then diffuse into the capillary bed, kind of in this uniform fashion, and then exit through the venule. However, in a disease vasculature that is hyperemic, meaning very high blood flows, you get this heterogeneous microvascular delivery. So in these slow flow, high extraction regions, you get slow flow, very high oxygen extraction. However, in these larger conduit vessels, or shunts, if you will, you can get this kind of low extraction, very high blood flow. And these vessels, these shunting vessels, contribute disproportionately to the venous signal, which will mean that what you observe is this kind of local mismatch between global oxygen supply and metabolic consumption on a very microvascular level. So of course, we're trying to explore ways to validate that further, but I think that's a very interesting finding. And then I guess in the last minutes I have or so, I'd like to talk about future applications. So we know that functional MRI has had a huge impact on a number of fields, including psychiatry, neuroscience, and just clinical neurology. It's one of the most cited techniques that come out of MRI. And if you look at this graph we're showing here, it's just, I did a Google Scholar search of brain and fMRI. And you can see that by 2015, we're peaking at over 40,000 publications per year for those two words. However, there's no real reason why fMRI is restricted to the brain. Well, there are some reasons, but there's no real good reason why it's restricted to the brain. Why can't we understand how other organs in the body are functioning with this kind of neurovascular or vascular coupling? So now here I'm showing in these yellow bars I put in the search kidney and fMRI. And you see it's much smaller, like almost it's insignificant compared to brain fMRI. And I would imagine many of these studies are not actually looking at kidney itself in terms of fMRI. But that's where I would like to go in the future. And it's not just a pet project. So sickle cell disease patients actually, they have a lot of kidney disease. So about 40% of patients with sickle cell disease have chronic kidney disease right now. And the median age of onset is 30 years old. So I know to grad students, 30-year-olds might seem old, but that's extraordinarily young to develop a chronic illness that is going to stay with you for life and most certainly contribute to early mortality. So about 16% of patients will actually die from chronic kidney disease. Additionally, it's just been understudied in pediatric disease. So it's an accepted fact that patients with sickle cell disease that are children all have kidney disease because they have problems concentrating their urine. It's just a ubiquitous fact. And then to put numbers to that, about 20% of 12-year-olds will actually have overt signs of chronic kidney disease, that is incredible, that there's really no treatment strategies that are really being used to prevent kidney disease in these patients. But like we talked about, we know that we can use fMRI to study brain kind of vascular coupling, but why can't we do that with the kidney? So I think we can. So if we can use the same model, so instead of using neurovascular activity or challenges, we can use challenges specific to the kidneys. So here we can use things like water loading or blood pressure challenges, salt loading challenges. And then we can look at the functional unit of the kidney, which is a nephron, and see how the vascular coupling is responding. And of course, we can now use fMRI or MRI to show these techniques or to show these vascular responses. However, it's an open question as to what the activity or challenge should be. It's also an open question of what the vascular response should be, as well as what techniques should we be using here? Because things that worked in the brain should not just be blindly applied to the kidney. I think that is not an excellent way of doing a science. So in order to have more kidney-specific tailored approaches, here I'm showing some work that I've currently been working on multi-dimensional oximetry in the body. So here is what a multi-echo radial sequence would look like. And the great thing about these continuous acquisition multi-echo radial approaches is we can retrospectively bin the data into all of these different dimensional states. So for instance, here I'm showing inspiratory and expiratory respiratory phases, but we can also just resolve things in time in order to get a respiratory signal. There's a lot of flexibility with these continuous acquisition strategies, which allows us to do free breathing, kind of whole kidney bold imaging. And some of the preliminary data is just showing that if I do a water loading challenge where I give healthy subjects 20 milliliters of water per kilogram, which equates to about a liter and a half of water, we can see vascular responses in the kidney. So here we're showing that you can see over time, the T2 star in the kidney seems to be increasing, particularly in what seems to be the cortex here, and as well as the collecting system. Also, if we look at phase contrast measure blood flow in the renal artery, we can also see that seems to be increasing. So this is where I would really like to take this. I think all of the gains that we made in the brain can now be applied to the kidney if we really are strategic about how we use these new techniques. So as a recap, I think I was able to talk about several things about how we can use translational imaging to really improve patient outcomes, historical examples, kind of just value add examples, and then kind of future applications that can really have profound impacts on patient management. And as a new professor at the University of Texas, it's currently just me right now, but I'm actively recruiting. So if you know anyone that's interested in kind of answering these type of questions, I'm developing an all-star team to really be able to tackle some of these challenges and really bring in the next generation of MRI imaging to the body. So with that, I'd like to thank all the people, past, present, and future, but particularly my colleagues at Stanford and Children's Hospital Los Angeles. So thank you guys for your attention.
Thank you so much, Dr. Bush. That was a really interesting talk. We can go ahead and open the floor for questions. Feel free to type in the chat with your question, or go ahead and unmute and we'll have a discussion.
Should I stop sharing, do you think?
Yeah, if you'd like to, you can see everybody better.
I'll start off with a question about kind of complementary imaging techniques to the BOLD measurement. And I'm really fascinated by the vascular shunt model and the possibility of the microvascular network being heterogeneous and kind of discovering where those heterogeneities lie. Are you doing any direct vascular imaging for morphology or blood flow in the brain or kidney?
We're not. So you mean like histology potentially or something? like that, or?
Even angiography techniques or like phase contrast MRI, just curious if that would be a good complementary technique to, and if you're really, thinking to pursue like the vascular imaging alongside your T2 star measures.
Yeah, we're certainly using things like phase contrast. The problem with the shunting model is like, this is probably happening on a microscopic level. So in order to image, we'd have to like take very small samples and probably would have to be invasive techniques. So, but the thing is, we are looking at other ways of kind of getting at the same information. So there's been actually some data with ASL showing that there's ASL signal that is now in the sagittal sinus in these patients, which might lead evidence that there is shunting going on. And several groups have identified that. There's also other techniques you can use, like optical techniques that can kind of, they're all different ways of probing the elephant. Like, and if we're able to, I think the great thing about just one paper, it's where is the evidence leading us? And if all the evidence is leading us towards shunting, I think then we can potentially begin to do some autopsy studies or some of these more kind of high risk studies that might be warranted.
Yeah, cool.
I have a question.
Hey, Nick.
Hello, that was inspiring.
Thank you.
My question for you is, you've shown trends that are meaningful with respect to the diseases that you're looking at with imaging, just with imaging. Sickle cell you can see in the blood itself. I'm wondering if you've considered combining the results from imaging with minimally invasive techniques like a blood test or something small that could be sent to pathology.
See, you're a genius, Nick. So that's exactly where I'm going currently. So I've got an ultrasound device, so a butterfly that I purchased, because I think this multimodal approaches that we can now, it allows us to really, like to Shelley's point as well, it allows us to probe these questions on all different scales. And sometimes it's appropriate to do an MRI, sometimes it's not appropriate to do an MRI. So if we shouldn't always just blindly move to MRI. So optical techniques, ultrasound, I think could really, blood tests as well, could really begin to, give us the same information.
Thank you.
Yeah, especially for the kidney. The kidney, all of the meat is, because the kidney essentially is its own laboratory that is non-invasive. So we have urine samples that are coming out. You can test the blood, you can image it, you can do biopsies of it. It's like, that's where I think we're gonna get some really big gains in the next like 5-10 years.
Very cool. I was thinking about some of the transcranial ultrasound folks that are on this Zoom meeting or in our department, like just in terms of collaboration potential and all, what kind of scanners and vendors are you working with? Is there any potential for collaboration there?
Right, so I think certainly, I know my advisor was on Philips. At Stanford, I was at GE, and now I'm on Siemens. So there's always someone to work with. It doesn't have to be me, but I'm happy if just the more people that are working on these problems, because it just moves the needle forward.
Absolutely.
Hey, it's Gary Smith. I'm one of the nuclear medicine physicians here at Vanderbilt. Great talk, very inspiring. I agree. And mostly because I see some of these techniques that you're mentioning are now being used in things on clinical applications in throughout the body, cardiac, liver, kidney, all these diseases that we deal with. I guess my question is pretty simplistic. It's from a clinical standpoint, how long does it take to acquire these data sets, and where do you see that going in the future? If now it's taking an hour or whatever, do you see these coming down to very short acquisition times for some of your your acquisition protocols?
Right. So I think we talked about a lot of different techniques, and there's variable timescales, right? Some of them are single breath hold, some of them are 5 minutes. But I think it's what you're getting at is really important. So what is the clinical need? And I think the best way to approach that is to be able to integrate some of these techniques into clinical medicine to get these outcome variables, because without data and what is the really the technique you need to be measuring, it's really hard to say, are all the techniques necessary, or do I just need to do one trust in the brain that takes a minute to get all the information I need? So this is where having your neighborhood radiologist or cardiologist on board with your research is really important to really get at what is the salient clinical information. And I think currently we don't really know.
Yeah. Thank you. Fantastic.
So, yeah. You might be muted.
Hi, you could hear me. Okay. So I thought that was really nice how you went through kind of the practical process of getting to clinical translation. And one of the things that I kind of always have a question mark about with new acquisition schemes. So asking about the Rosette, if you can talk a little bit about the reconstruction time and kind of the computational power you would need for it. And then how do you go about setting up or having conversations with clinicians to set them up to be able to do that? Or do you have to go directly to vendors, et cetera?
Yeah, it's a great question, right? Because at Stanford, all this is done at Stanford, all the non-Cartesian work, which has 20, 30 years of just like infrastructure built. And that's not at every place. So I understand that's a real challenge. But that said, vendors are on board for this already. Vendors already offer many non-Cartesian imaging strategies. Compressed sensing is a product package on many platforms now, I think all of the major vendors. Additionally, things like XD Grasp are product packages. So if you can have a killer app, the vendors can't wait to take a bite out of it, right? So that's our job, to give them the killer app. And then that just motivates them to get the work done as far as getting people to buy it.
Right.
And there's nothing wrong with, I think sometimes as scientists, we're like, oh, I don't want to like sell this thing, but sometimes that's how it gets out into the ether. It has to be sold. So,
Right.
So yeah, I think that is also an important thing. Talking to your vendors, making sure that we're speaking the same language, and we're trying to get these things out into the hands of real users that are seeing the patients.
I see. Okay, thanks.
Great. Are there any last questions for today? All right. Well, thank you so much again.